Now I'd like to introduce today's speaker. Today's featured speaker is Ada Poon. Ada Poon is an Associate Professor of Electrical Engineering and a Terman Fellow at Stanford University. Her research focuses on providing theoretical foundations and engineering platforms for realizing electronics that seamlessly integrate with the body. This is emphasized in her research group by using a vertical integration of diverse fields ranging from physics, wireless technologies, and low-power integrated circuits. She holds a PhD degree in Electrical Engineering and Computer Sciences from the University of California at Berkeley and received her Bachelor's of Engineering degree from the University of Hong Kong. She has received the Okawa Foundation Research Grant in 2010 and the NSF Career Award in 2013. So now I'd like to turn the floor over to Ada for her presentation today.
Thanks, Stephanie, and thanks for everyone joining the webinar. Before I start the presentation, I would like to just give a very brief introduction about my background. I study, as Stephanie mentioned, I study my PhD at Berkeley, and it is in the area of information theory for the application in the wireless domain. And after graduation, I spent some time at Intel to design radios, and also work in a startup to design high data rate radio, which is the predecessor of what we now know about L2.11ad. We try to accomplish gigabits of data rate. After a while in industry, I decided to go back to academic and totally change my research area from information theory to medical systems. And especially, what I would like to do is I would like to see if I can build electronics in the scale of a medicine pill, such that we will use the electronic method to treat diseases rather than a chemical method. For example, right now when we are sick, usually we will take a drug and eat a pill and hope that we will get better. And by taking a pill, here, we're using a chemical way of modifying the biological activities. But biological activities can be modified not only by a chemical way, but also can be modified by an electrical, thermal, mechanical, and the optical way. The reason why the chemical way dominates, if you're looking at the history of the pharmaceutical, actually 100 years ago, all the different methods, they're very, it's well used, all the different methods, not only the chemical way. The reason why chemical way dominate, it is because it is disposable. That means pharmaceutical companies can earn a lot of money by having patients to take the drug and then to keep buying the drug. But at the end of the day, not only the chemical, we can treat diseases not only through the chemical way. And in my research, I'm focused, it is on, because I'm in electrical engineering, my research is focused on using the electrical way and the optical way to modulate the biological activities to achieve a therapeutic treatment. In particular, I'm very interested to build devices that are very, very small, like this one, that I could put close to the nerve or to the organ where the therapeutic actions take place. And I do believe that by putting the devices close to where the action point is, we would have less side effects than taking drugs, because drugs act more globally throughout the body. And that's the mission and the vision that I had at the time when I returned to the academic. It is, I really want to build electronics or devices that are so small that it could really put close to the nerves or the organs for therapeutic actions. And in order to do that, one of the major obstacles to make these devices very small such that it can put close to the nerves or to the organ to take the action, it is the energy source. When we are talking about energy source, first of all, we think about using a battery. And currently, in most of the implantable devices, the battery occupies more than half of the space, and it is in the scale of a centimeter. And there are newer methods like piezoelectric, glucose harvesting, thermoelectric, and biopotential The problem with all these new methods is that the power density is far below 0.1 microwatt per millimeter square. Let's imagine that we need to consume about 10 microwatt of power in order to do something useful. Then the dimension of all these new methods, the source dimension will be on the order of centimeter. That is, we haven't solved the problem of the bulkiness of the battery. We just moved the bulkiness of the battery to the bulkiness of the other energy harvesting devices. As I mentioned before, my background, it is in wireless. And the reason why we can transmit data over the air, it is because we're able to transmit energy over the RF energy over the air. So how about we harvest the energy wirelessly, such that we can remove the battery entirely? And energy harvesting using the radio frequency signals is not something new. And actually, we're using this long time ago. The devices that on your left, it is a, you may wonder what it is. It is a pacemaker, the first pacemaker. And as you can see that inside the pacemaker, it is like a metal cane. There's a coil, this is the coil. And then it receives energy, it is also wirelessly from a device that it is put on the chest. So the mechanism is very similar to our electric toothbrush. You could assume that the pacemaker, it is like the toothbrush, and then the devices that put on the chest, it is like the base station for the toothbrush to get charging. And the mechanism for both are, I would say that it is pretty much the same. And the person who implant these devices, it is the first people who implant it, it is in 1958, which is more than half century ago. He implanted at the age of 43. After implanting these devices for a few hours, he has to implant another one. And at the end, he had 26 pacemakers got implanted into his body. And he died at the age of 86, not because of cardiovascular disease, but it's because of skin cancer. So 26 pacemakers, and the earliest one is the one shown here, using wireless powering to power up the pacemaker. And in the 1990s, this is the cochlear implant, which is using similar idea. It is like the electric toothbrush to charge up the devices. So this is the external device, this is the internal device. And as you can see that there's also a large coil that is on the implant, and because the coil is so large, it is just put under the skin, and then there is a neat wire here to connect to the electrode, and the electrode is putting inside the inner ear for the stimulation. Now what I want to do is I want to cut all these devices and the wire, and I just want to have that part, the electrode part, left. And this is my device, such that I could just implant it in the location where I want to do the stimulation. So in order to do that, the first question that I have in my mind is why everyone else using inductive coupling as a mechanism to deliver power wirelessly to the implantable devices, which is, as I mentioned, it is the same mechanism as the electric toothbrush. So why this is the dominator mechanism? What if I have a freedom to choose the source that I could put outside the body? Any shape, doesn't need to be a circular loop as the one that we used before. What will be the optimum dimension, optimum shape to deliver the maximum power to a small implant inside the body? So I started this problem in 2008 as a very theoretical problem. It is, I don't mind and I don't care how to implement it. I just want to obtain the upper bound. Once I obtain the upper bound, then the way to get the upper bound will give me intuition on how to realize the optimal source. And I have a way to justify whether the source is already good enough. without the upper bound, I don't know whether the source that I'm designing it is good enough. And the design method, the way I formulating the problem, it is very similar to the way we solve problems in information theory. For example, in information theory, we will try to find out what it is the channel capacity without talking about how to implement the coding and decoding algorithms in order to achieve the capacity. But I'm not going to go into the detail of the mathematics involved, but instead I would like to show you the results. So luckily, after solving the Maxwell equations, we are able to obtain the solution for the optimal source and the optimal efficiency. As I mentioned, I don't want to go into the math. Instead, I will show you a numerical example where there's the optimal source current density is trying to power up a device that it is put on the heart across the chest wall, which composed of 1 centimeter air gap and 4 centimeter heterogeneous tissue, for example, skin, fat, muscle, bone, rib bones, and then heart muscle. The graph, it shows that the efficiency at about the 2 gigahertz range, the efficiency, it is 2 orders more than the one that we currently use, that it is in this region, in the inductive coupling region. And the reason why we can accomplish such a high efficiency in the optimal source is because, let's looking at the circular source that we used to use in inductive coupling, like the electric toothbrush or the early pacemaker, and also the optimal source structure, which is given in the figure here. When we're looking at the loop source, the magnetic field that it is from this source is divergent. As you could see from the Poynting vector, the white line, it diverges. But the magnetic field from the optimal source, it converges, it tries to focus to the location of the implant, and then after focusing to it, it diverges. And because of this focusing effect and also the redistribution of the hotspot, which is the loss corresponding to the loss, that's the reason why we're able to get about 2 orders of magnitude better efficiency than the current solution, which is based on the inductive coupling. Let's take a closer look on what happened from the optimal source. For the optimal source, what happened is the electromagnetic field, the magnetic field that is outside the close to the source and outside the tissue, it is evanescent in nature. That is, it is non-propagating. But when it interacts with the tissue, it becomes propagating. As you could see, that majority of the wave components is within the dotted circle, which is the wave number. Within it, it means that it is propagating. Outside it, it means that it's non-propagating. Now, this is very different from inductive coupling, where the magnetic field outside the source is non-propagating. When it interacts with the tissue, it is also non-propagating. And that's the reason why the inductive coupling method or the near-field method, the magnetic field has limited penetration, because it's non-propagating outside and it's also non-propagating inside. While the optimal source, it is non-propagating outside, but it becomes propagating inside the tissue when it interacts with the tissue. And this is the fundamental difference between the optimal solution versus the one that we conventionally use. And because of this property, we are able to make the devices very small and implant it pretty deep inside the body. Now, in order to, so far we talk about just a mathematical solution and a mathematical result to show that what is the optimal way to transmit energy across a set of heterogeneous tissue. In order to implement this source, we found out that the source, when the tissue is removed, it is not propagating. When there's a piece of tissue there, the field is propagating. And this is not something common with an antenna structure. Initially, we talked to some antenna people. We tried to get help from them, but they have problem understanding our requirements because it is hard for them to decide a source that is non-propagating. As the one you see here, when there's no tissue, it is not propagating, then it is not antenna anymore. And they are true, this is not an antenna. And because of this special property, we have no choice but buy the Poon ourselves. And I spent half a year trying to learn the electromagnetic tools, and every day just looking at the optimal current distribution that's shown on your left for half a year, and try to find a source that will mimic this optimal current distribution. And luckily, we are able to find a source as seen on your right, that it is composed of some circular slot structures and some circular loop. And with this structure, we could accomplish about 10% from the optimal solution, as you could see from this curve on the receive power versus the source device separation. In this experiment, it is building the coil that is about 2 millimeters in diameter and 3 millimeters long. And in order to measure the receive power, any wire that attached to this coil will interfere with the radio frequency. That is, the measurement that we got will be inaccurate. Most of the energy will be picked up by the cable. And in order to solve this problem, we built a tiny circuit where it will harvest the energy from this tiny coil, 2 millimeter coil, and then there's a circuit that will convert this energy into pulses. And the frequency of the pulses is proportional to the received energy. By adjusting the different level of the input energy and attaching an optical fiber to an LED. So optical fiber will not interfere with the radio frequency transmission. Therefore, it solves the problem that the cable will pick up the energy instead of the coil. By measuring the frequency of the light pulses, we are able to infer the receive power in this experiment as shown in the curve that on your right. So after we have this source, we put the probe that I showed in the previous slide, and into a 119-pound tape, and putting in the brain, and also putting in the heart, and also in the brain of this Pochi model. And with the transmitter, which is emitting about 500 milliwatt of power. So the reason why I pay 500 milliwatt of power is because this is pretty much the average output power of a cell phone. So if I use this number, it should be safe. It is able to deliver about 200 microwatt of power into the heart and into the head, which is about 5 to 5.5 centimeters from the surface of the animal. It's quite deep. And the devices that we're using is only 2 millimeters in diameter. And in order to put it into perspective, what 200 microwatt of power can do, our pacemaker consumes about 10 microwatt, and deep brain stimulation consumes about 100 microwatt. So with a source that outputs 500 milliwatt of power, which is pretty much the same output power of the cell phone, and the device that we are using is also on the dimension of the cell phone, which is 6 centimeters, which is about the size of the iPhone 5, not the 6S. 6S is too large. As it's able to deliver enough power for both pacing and deep brain stimulation with these devices, that it is quite tiny. Now at this point, we got a lot of criticism from people saying that, is it safe? Even though I tried to make it the transmit power to be 500 milliwatt, which is the average cell phone emitted power from the cell phone. So if you can accept cell phone is safe, then you should be able to accept that our device is safe. But since we got a lot of criticism from people, and before I joined Stanford, or before I come back to academic, I worked at early stage startup. And the way we try to certify our devices is to go to an independent lab and then certify safety. So I use the same approach. I just want people to shut up. And then we send our devices to a certification lab to demonstrate that the device is safe. And to my surprise, Okay, before we got the report back, I just feel that this is an academic project. If it is barely meet the safety threshold, I'm pretty happy. But to our surprise, it is 10 times below the safety threshold. That is, it is really safe. And this is the setup that the lab is doing in order to measure the safety of the devices. Basically, the device is put underneath a tank of solution that mimics the body, and there's a robot that is measuring the electric field in the solution point by point, and then find out what's the P-specific absorption rate location, and then do the processing and give us the report back. When I got the report and I find out that, wow, it is quite safe, I try to understand why it is so safe. So we go back to the source that we decide. And as you could see that in our source, we fit the devices into 16 locations. There's 4 ports, but then it actually has 15 locations to fit the device, to fit the source. And the hotspot, the hotspot usually it is close to the feed line, close to the feed region. And here we distribute the feed region to 16 locations. And that's the reason why our size it is pretty low. It is 10 times below the safety threshold. It is by accident when we decide the source, we distribute the feed regions into 16 locations. Now, at that point, we become more ambitious. We say, okay, let's increase the transmit power such that we barely meet the safety threshold. In this case, we are able to deliver about 2 milliwatt of power at a distance of about 5 centimeters to a device that's only 2 millimeters in diameter. So 2 milliwatt of power can do a lot of things. Currently, we're trying to integrate it together with a camera that it is 1 millimeter cube in dimension. And imagine that, you know, with a 256 by 256 pixel, 14 frames per second. We don't know what it's going to be useful for, but we feel that it is pretty cool to do, you know, a tiny camera that you could look around inside your body to do something interesting. So after we demonstrate the safety, we also demonstrate the real-time tracking. Now, I don't want the patient who holds the transmitter has to keep moving it in order to find the spot that will deliver energy optimally to the implant inside. This is not user-friendly. I would like to accomplish this electronically. And this is very similar to when we are taking pictures for the camera. Using the camera, we do the auto-focusing. And in these devices, we are also able to do the auto-focusing, as you could see from the panel or graph that on your left, upper left, that it shows it could focus the energy to the different steps and the different directions. And in this example, we use a Stanford S to illustrate these properties. When adaptation is on, the entire Stanford S light up when the device is moving, you know, within the source region. When the adaptation is off, only those points that it is near the focal point of the source light up. And the coverage region, you know, when adaptation is on, it is about 6 centimeters. That is, if the patient, you know, holding the devices that it is offset by about, you know, a few centimeters, we are still able to correct it. So now we demonstrate the theory behind and then try to realize the source to approach the optimal solution, and then demonstrate the efficiency, safety, and also real-time checking. Last thing is we want to demonstrate that it could do something. Now we move in the LED, and then we just put 2 electrodes into the same device. It becomes a pacemaker. So pacemaker is really easy to build if you want to build one. In here, it shows the pacemaker that it is on a put on the finger. We find a student that with the biggest finger, such that it could get a pretty good contrast. But we also put it close to a catheter to illustrate that it could be delivered, you know, through a catheter using minimally invasive procedures into the body. In this case, we put the devices using open chest operation into a rabbit. It is the first time I see a physical rabbit. I think, I didn't know that rabbit is so large. It is more like a bunny to me. And then it is very cute bunny, and we have to open the chest and stick it in the devices into applets of the bunny, and then close the chest and then turn on our source. Here it shows that when the device is on, when the source is on, the pacing of the rabbit, it is more regular and faster to illustrate the capability of accomplished cardiac pacing. So afterwards, being a theorist by training, as I mentioned, and my PhD is in information theory, the way we decide the source actually is quite ad hoc. I look at the source, and then I will try to find a structure that will mimic the optimal source. As I mentioned before, the interesting thing about the source is that when the ways that it is outside the tissue is non-propagating, when it interacts with the tissue, it becomes propagating. It needs me to think about the generalized Slaoui's law. So I hope that a lot of you will be familiar with Slaoui's law. If it is not, then let's look at this photo. This is the Slaoui window, which is the 180 degree of the world as seen by a fish that is under the water. It becomes about 100 degrees, and outside this 100 degrees it becomes dark. So what happened? This is because of the refraction, that when there's a wave that is incident from air into a material, the ray will bend, right? Because it's different permittivity, different index. And there's a certain angle where it cannot go beyond inside the material, and this is called the critical angle. Now, what we want to do in our optimal source, indeed, it is we want to reach the forbidden regions. That is, we want to reach beyond the critical angle. Now, this can be done currently by using immersion lens, but you don't want the patient to hold a 3D structure outside the body in order to bend the waves. Now, what we actually doing with our optimal source, it interacts with the incident field. When it interacting with it, it makes it to be able to reach the forbidden regions. That is, go beyond the critical angles. Now you may ask why I would like to do that, why I would like to reach beyond the critical angles. Here, I would like to use time and frequency to explain this phenomenon. In time and frequency, when we have a signal that it is a very wide band, the signal in time domain will be very narrow, a very narrow pulses. Similarly, in here, if I can generate a signal that has a very wide angular spectrum, that is, if it can reach the forbidden regions, forbidden angles, then it will have a very wide angular spectrum. Equivalently, there will be a very narrow spatial signal, and that's the reason why you see that there's a focusing effect in the optimal source. We basically doing sub-wavelength focusing to achieve a high power transfer efficiency. And in order to accomplish that, we are very lucky. The structure actually is quite simple. It is made of some metal strips, but each of them it is sub-wavelength spacing. For example, in this case, in this example, we use the metal strips that within one wavelength we place 20 metal strips, and each strips are noded by a different passive element. By adjusting the elements, we are able to launch the wave that it is with the refracted angle, it is greater than the incident angle. It means that we already go beyond the critical angle, and we are able to reach the forbidden region that is beyond the critical angle consistently with the different incident angles. It also confirmed that without the material, without the tissue, no propagation, which is very similar to the optimal solution that we showed before. And by superposition, we are able to generate the angular spectrum, the waves that is able to do sub-wavelength focusing, as you see in this slide, that we're able to accomplish 1/8 of the lambda in the focusing into the material. And the structure that we're using, it is a ring structure and then loaded by different passive elements. And finally, we use these devices. This is the structure that we're using. The dimension is about 6 centimeter, 6 centimeter in the width, and this is our transmitter and the battery. Now we are holding these devices, which is quite small, to do a hard pacing for a pig. So in this case, we don't do open chest anymore, but instead we deliver the pacemaker with the minimally invasive procedures by drawing a hole in the thigh of the pig or in the leg of the pig, and then deliver the pacemaker using a catheter. And we put it inside the right atrium, the right ventricle, and also the left ventricle. Here it shows the X-ray images of the devices after implantation, and also the location of our transmitter that put outside the chest, put on the skin of the animal. What we want to demonstrate is we want to put the devices in the different locations of the heart and to do a more synchronized pacing. So here you see that when the device is on, it does the pacing in the different regions, and off, then the heart rate is slower. And when it's on, the heart rate it is faster, and there's a certain lagging certain delay when the device is on. There's a certain delay before the heart rate increases. The power for the left ventricle, the required transmit power, it is higher, about 200 milliwatt, because the wall it is much thicker than those that it is in the right ventricle and the right atrium. In the right atrium, the transmit power, this is the transmit power from the transmitter outside, it's about 34 milliwatt. So here we demonstrate that with the pacemaker, with minimal invasive procedures, we are able to use our source, which is a very handy handheld. We can even make it flexible, very easy to use source to do the pacing. And this is the pacemaker that we put in the that compare with the penny. A lot of time people thought that the silver part is the antenna. It is not the antenna. The silver part is just for fixation, for anchoring. The antenna, it is the red coil here, the 2-turn coil. And then there's the electronics that is putting inside, and one more electrode, it is inside the silver coil. This is the return electrode, and then at the tip of this silver coil, it is another electrode. And then all the other parts coded, such that the 2 electrodes and the electronics put with the code opposite to code it. So here, so far we just talked about pacemaker. But as I mentioned at the very beginning, I want to make electronics that are small enough such that it could put close to the nerve or the organ to do some therapeutic treatments. Pacemaker, it is not what I want to do, but it is a very good way to demonstrate the feasibility. In order to move on to our original ideas of the bioelectronics medicine, we want to do something that is to show this idea. In order to do that, I look at the neural pathways, the 12 nerves that it is from the brainstem, and eventually I picked number 10, which is the vagus nerve. At the time when I returned to academia, I always heard people talk about vagus nerve. So I think this is a magic nerve. It can be useful in anything, seems to me. So if I pick this nerve, there may be something that will happen. And in particularly, I focus on heart failure. So basically, I do vagus nerve stimulation in the right vagus nerve stimulation in the pig model for heart failure treatment. In here, this is our transmitter. Now it becomes banded with the adhesive such that it could be just stick it into the leg of the patient. And then this is the receiver that is put on the vagus nerve. And the transmitter and everything are integrated in this cup electrodes. And here it shows the X-ray images where we implanted the devices, and it shows the effect. of the input power versus the heart rate and also the blood pressure. Okay, and so we say about 20 milliwatt of input power from the source that is on the adhesive, we're able to change the heart rate by 20%. Similarly, with the input power of about 20 to 30 milliwatt, we're able to change the blood pressure by about 20 to 30%. And here this graph shows, that we can consistently get it from, this process is reversible. That is, when we turn on the devices, the heart rate drops. When we turn it off, heart rate rises again, and turn it on, heart rate drops. Similarly for the blood pressure. And then here it shows the dynamic of it. When we have a pulses that it is 10 minutes on, 10 minutes off, you see that the heart rate, it is pretty much follow the pattern of the on and off. For the blood pressure, it takes longer time to get to the initial stage. Therefore, the blood pressure keep decreasing. So if we increase the interval to be, for example, half a minute, we may be, it may be able to show what we see for the heart rate. But anyway, this demonstrates the feasibility that one application could be in hypertension. Instead of taking the drug to reduce the blood pressure, now you put a patch and then on the neck, and then by delivering the electrical pulses, it could reduce the pressure. So it cannot demonstrate the feasibility of using electronics to replace drugs. In this case, we are targeting it is the physiological state is the blood pressure. Going even further, so far we use the electronics, it is to stimulate the nerve bundles. But it would be nice if we can stimulate the individual axon. And here it goes to the technology of optogenetics. So using light, so far what we're talking about is electrical stimulation. Electrical stimulation is lack of the cell specificity, while optical stimulation using light, we're able to target to a particular neuron type. So that's the differences. And the current light delivering systems, this is the one that used in our collaborators lab, Daisaro's lab, that's the optical fiber, and then it delivers the light into the animal. And these two are the wireless versions. One it is from Japan, another it is from MIT. It's very large headstand that is putting in the on the animal. So imagine that you have the same volume and weight of your head that is put on top of your head. I don't think you will feel that this is freely behaving. But this is what happened to the animal experiments while they're trying to use the wireless light delivering systems. Another, with this kind of devices that it is for the MIT one, it is also wireless power. Therefore, you could see that there's a coil array here, the red one, many terms. Now, in order to increase the area where the animal can walk around, researchers, in my opinion, it is a little bit crazy. They put a magnetic sensor into the animal feet, and then they try to track the location of the animal. After tracking where they are, they turn the coil that is underneath the animal. So basically, you could see that this is the smallest cellular cell in a cellular network, and the customer, it is a single mouse. And then there will be long overlapping cells or overlapping cells. Whenever, imagine that whenever you go anywhere, they will locate where you are and then turn on that cell. And this is what happened to this case. But instead of the human being, it is the animal. There's another system that is developed by EPFL. Instead of having multiple cells and turn on the one that's close to the animal, they have the cells being moving with the animal, and then track where the animal it is and keep powering it. Now I think this is too much work, and when I encounter this problem that we need to increase the propagation range of the system such that the animal can be freely moving in a larger area, I feel this is crazy, and I want to be lazy. And in order to be lazy, we need to be a little bit more creative. So being inspired by quantum tunneling, we developed a self-tracking system that doesn't require any electronics, so we can be lazy. Okay, let me play the video to illustrate how it works.
A microwave cavity is a hollow metal structure. When it is excited by an RF source, it confines electromagnetic fields inside it. If we remove part of the metal structure, for example the top plate, energy from the source will radiate. But if we cover it with a metal plate patterned with sub-wavelength apertures, the honeycomb, energy from the source will confine within the hollow structure. There is no radiation. However, when a mouse is placed on the honeycomb, energy will be transferred to the mouse through the fringing evanescent field. This energy transfer follows the mouse over any path of motion on the honeycomb, achieving self-tracking without any tracking mechanisms.
We make use of the properties that animal, the tissue of the animal, basically we are made of 70% of water, right? The dielectric properties of water is about 80 times larger than the dielectric properties of air, permittivity. And we make use of differences in the permittivity to do the self-tracking as explained in the video. And in order to do that, then we need to study the resonant modes of a small animal. I think this is the most ridiculous thing I ever done in my academic career. It is to study how a mouse resonates. And this involves the drawing of a very cute mouse in the electromagnetic simulator, and then scale it small and large, and then with different orientation to figure out what are the resonant modes are, and such that we can design our cavity around that resonant frequencies. And after we decide the cavity, which is where it can do self-tracking, the animal can run freely onto a larger area. Next, we need to build the implant. So here it is the devices that we build that to put in the right motor cortex of the animal. We follow the same implantation procedures as the one that they did my collaborators did in the for the optical fiber one. Therefore, we have to attach the optical fiber for holding. Afterwards, we break the optical fiber and then do the citrate. So at the end, you will see that the whole implantable devices, it is fully inside the body. There's nothing that it is like the head stage. Here you see the light coming out is because when we write papers, we cannot say that the animal already has an implant inside. No one will believe you. So therefore, we have to increase the power a lot such that the light is lit out from the scalp, such that you will see that's the light. But in reality, it will be something like in this video that I want to show what happened. So we put the implant into the right motor cortex. You will expect that when the stimulation is on, the animal will move in counterclockwise. In the left, has the tendency to move left. So it will move counterclockwise in making circles. And as you could see that the animals look normal, right? You don't see an implant, but it's already there's an implant inside to control its brain. So you see that's the circling behavior. The animal doesn't want to move in the counterclockwise direction, but it is forced to move. We're basically controlling the mouse. So here you see that when the stimulation is off, it's random. When the stimulation is on, it do the circling effect. Okay. So let me conclude this talk. We talk about electrical stimulation. We also talk about optical stimulation. And at the end, we really hope that we can build the devices as small as the pill, and you will get this kind of devices in the countertop in the drugstore instead of the chemical one. And that concludes my talk.
Great. Thanks so much, Ada. So if you haven't submitted your questions, please do so as well. But Ada's going to choose some questions to go ahead and tackle now.
So the first question is, how specialized would manufacturer be, or could existing medical device manufacturers make this? I would like to say that the technology that I just described will be licensed to 2 startups and also 1 big medical device companies. They are translating them into something that it is useful. So therefore, I would say that existing medical device manufacturers could make it. And to my best knowledge, that the focus of those startups are what I heard from the technology office. It is one, it is for pain relief. Another, it is for urinary incontinence. So if this FDA approved air security, so I would let the 2 startups and the medical device companies to show it, because they pay money to get it, right? They better show that it could be FDA approved and pass the airport security. What kind of health problem is this application focusing on? As I mentioned, right, the 2 companies that licensing our technology, one is focused on pain relief, another is urinary incontinence, but they may also work on other applications. Those are the applications that I heard from the technology office. The longest that the question is, will the device have any immune response, or is there any information when the device is inserted in the host? Now, so far we only do animal testing. The longest one that we did, it is about 1 month, and it seems fine. When we exploit the devices out, and then it seems, and do the histology, and it seems the surrounding tissue, it looks fine. So is it biodegradable, how stable devices in the body? As I just mentioned, right, the longest we did in animals is 1 month. And I will also let the 2 startups and other companies, you know, they work on the biocompatible problems. These devices have proof, and will it require a proof to? Yeah, of course, it is not have proof. We need to. It is just a stimulator that with wireless control. It needs to have more functionalities like a processor in order to do the encryption and decryption to make it to be hacker-free. Okay, based on the energy transfer in positive models, are changes to the process of device required for different body mass? This is a very good question. Now, before we do the animal testing, we kind of worry that the structure will affect how the device performs. So we have a lot of backup solutions. But at the end, when we do the animal testing, we realize that they're pretty much the same. We don't need to adjust the device for the different animals. So I would say that it doesn't need to change, because the variation, it is not too much. You see this as being able to give continuous feedback from neurological systems. So the question is, do you see this as being able to give continuous feedback from neurological systems? This is the current problems we are solving. We're trying to incorporate both a neural recorder and the stimulators, because we have a wireless transceiver already built in silicon. We're also collaborating with others to build the electrical recorders with a few hundred channels and stimulators to do the neurological applications. How do you remove these devices when the delivery or use is completed? This is a very good question. Usually after we finish the experiment, we just kill the animal. We remove it. But of course, in order to do that, we need to really, decide some special catheter that is able to deliver it, and at the same time, it's able to unscrew it and remove it. What is the lifespan of these devices? What is the lifespan of these devices? It depends on the coating. For the one that we do accurate study, we just use regular epoxy, so it doesn't last for a long time, maybe a few days. But for the one that we try to do the more chronic study, we need to send it out to outside companies to do special coating such that it could last for much longer. If taken in pill form, how will the device go to the location in the body where it is needed? So for if you're looking at the devices, even though we put it in the pill form, there's some anchor, several nicks that is trying to anchor them into the tissue or into the nerve. Implantation is always a challenge for medical devices, and therefore it requires a lot of, it will be application specific. The way to implant the devices, it will be really specialized to the type of applications that we are targeting at. So I cannot answer this question, you know.
So it's 11, and we want to be thoughtful of people's time. So maybe, Ada, you can pick 2 more questions to answer, and then we'll wrap it up for the day.
Okay, can you explain the critical angle more? So critical angle, basically, when we have a ray that it is emitting from air into water, for example, you will see that the ray, for example, you shine the light from outside the water to the water, inside the water, you will see that the light bends. And then it is bent towards the center. And there's a certain critical angles where the light will not pass it when it is inside the water. And this is the critical angle. And that's called the Slaoui window, as I show in the photo. That's the reason why the 180 degree of the well, that it becomes only about 100 degrees from the fish that is inside the body, inside the water. It's because of this critical angle effect. Do you have any plans to extend research into studying these applications to a wireless 4D area network? Actually, I don't really buy the idea of wireless 4D area network, so no.
Great. Okay, so we are going to wrap it up. I want to say a big thank you to Ada for a wonderful presentation. Your research and what you do is so exciting, and for taking time to answer attendee questions as well. And we want to thank you for taking the time to attend today, and we'll see you at our next webinar. Thanks so much, and have a great day.
Thank you. Side effects than taking drugs, because drugs act more globally throughout the body. And that's the mission and the vision that I had at the time when I returned to the academic. It is, I really want to build electronics or devices that are so small that it could really put close to the nerves or the organs for therapeutic action. And in order to do that, one of the major obstacles to make these devices very small such that it can put close to the nerves or to the organ to take the action, it is the energy source. When we are talking about energy source, first of all, we think about using a battery. And currently, in most of the implantable devices, the battery occupies more than half of the space, and it is in the scale of a centimeter. And there are newer methods like piezoelectric, glucose harvesting, thermoelectric, and biopotential. The problem with all these new methods is that the power density is far below 0.1 microwatt per millimeter square. Let's imagine that we need to consume about 10 microwatt of power in order to do something useful. Then the dimension of all these new methods, the source dimension will be on the order of centimeter.
Now I'd like to introduce today's speaker. Today's featured speaker is Ada Poon. Ada Poon is an Associate Professor of Electrical Engineering and a Terman Fellow at Stanford University. Her research focuses on providing theoretical foundations and engineering platforms for realizing electronics that seamlessly integrate with the body. This is emphasized in her research group by using a vertical integration of diverse fields ranging from physics, wireless technologies, and low-power integrated circuits. She holds a PhD degree in electrical engineering and computer sciences from the University of California at Berkeley and received her bachelor's of engineering degree from the University of Hong Kong. She has received the Ocala Foundation Research Grant in 2010 and the NSF Career Award in 2013. So now I'd like to turn the floor over to Ada for her presentation today.
Thanks, Stephanie, and thanks for everyone joining the webinar. Before I start the presentation, I would like to just give a very brief introduction about my background. I study, as Stephanie mentioned, I study my PhD at Berkeley, and it is in the area of information theory for the application in the wireless domain. And after graduation, I spent some time at Intel to design radios, and also work in a startup to design high data rate radio, which is the predecessor of what we now know about L2.11ad, which has accomplished gigabits of data rate. After a while in industry, I decided to go back to academic and totally change my research area from information theory to medical systems. And especially, what I would like to do is I would like to see if I can build electronics in the scale of a medicine pill, such that we will use the electronic method to treat diseases rather than a chemical method. For example, right now when we are sick, usually we will take a drug and eat a pill and hope that we will get better. And by taking a pill, here, we are using a chemical way of modifying the biological activities. But biological activities can be modified not only by a chemical way, but also can be modified by an electrical, thermal, mechanical, and the optical way. The reason why the chemical way dominates, if you're looking at the history of the pharmaceutical, actually 100 years ago, all the different methods, they are very, it's well used, all the different methods, not only the chemical way. The reason why chemical way dominate, it is because it is disposable. That means pharmaceutical companies can earn a lot of money by having patients to take the drug and then to keep buying the drug. But at the end of the day, not only the chemical, we can treat diseases not only through the chemical way. And in my research, I'm focused, it is on, because I'm in electrical engineering, my research is focused on using the electrical way and the optical way to modulate the biological activities to achieve a therapeutic treatment. In particular, I'm very interested to build devices that are very, very small, like this one, that I could put close to the nerve or to the organ where the therapeutic actions take place. And I do believe that by putting the devices close to where the action point is, we would have less, that is, we haven't solved the problem of the bulkiness of the battery. We just move the bulkiness of the battery to the bulkiness of the other energy harvesting devices. As I mentioned before, my background, it is in wireless. And the reason why we can transmit data over the air, it is because we're able to transmit energy over the RF energy over the air. So how about we harvest the energy wirelessly, such that we can remove the battery entirely? And energy harvesting using the radio frequency signals is not something new. And actually, we're using this long time ago. The devices that on your left, it is a, you may wonder what it is. It is a pacemaker, the first pacemaker. And as you can see that inside the pacemaker, it is like a metal cane. There's a coil, this is the coil. And then it receives energy, it is also wirelessly from a device that it is put on the chest. So the mechanism is very similar to our electric toothbrush. You could assume that the pacemaker, it is